Holy f***ing shit. Good evening, Pittsburgh and universe. This is JC on a bike. My name is JC and this is Journal Club on a Bike. Tonight we're covering a paper by the Cohen lab, chemistry department at Harvard. A guy who's been working for quite a while on genetically encoded voltage indicators as a way of recording electrophysiology or measuring electrophysiological signals in cells without pipettes. Thank you. And so this has been a goal of Michael Hauser and Fritjof Helmchen. Lots of researchers have been working for many years to try and get all optical physiology to work. In slices you could do targeted whole cell patching of connected neurons. You could look at inputs to dendrites. There are all kinds of experiments that you could do if we had a way of accurately measuring voltage locally in a neuron. And so genetically encoded voltage indicators are where it's at for this. The construct that they're using here uses a voltage indicator called Archon. And this is a soma-centered archon expression construct so that they can image the voltage of the soma. And so in the first figure, they're just going to show us how this works and whether it works or not. And so they're going to show you stimulation and inhibition with the combination of these two tools. The tools are either the voltage indicator and a channel rhodopsin, or the voltage indicator and a hyperpolarizing current channel. I don't know off the top of my head which hyperpolarizing current channel they use, but they use a hyperpolarizing current channel to do the inhibitory version, the I-optopatch, and then the other one I think is optopatch 4. So in this first figure, they're just going to show you that, number one, they can make the recordings and what those recordings look like. And then they're also going to show you that they can use the channel rhodopsin to depolarize the neurons and to get them to fire, and that they can resolve single action potentials in the recordings. And then they're also going to show you that they can inhibit the cells and what that looks like. And so it's basically, the first figure is just basically proof of principle of the idea that they can optically monitor voltage while also stimulating, which is required if you want to do really, call it patch clamp or all optical physiology. Then you need to be able to control current injection. And so that's the combination with the opsins. It's a nice figure. Key part of this figure is to notice that the imaging that they're doing with the highest signal to noise ratio is imaging that uses holographic projection to excite only the membrane rather than the entire soma or the entire wide field view of the microscope. And they show in this figure how much more signal to noise they get with this special kind of holographic illumination, and it's quite a change. They also show in this figure that depth really counts against Archon. They lose their signal to noise very rapidly as they move deeper into the cortex. And that gets back to the point of, why are they doing layer 1? he's in the chemistry department and he doesn't really care. So they chose layer 1 most likely because it's the most accessible, it's the easiest to image, and you know, there was a low-hanging fruit there. You definitely couldn't do layer 5 pyramidal cells with this prep, and they know that, of course. So they're looking at layer 1 interneurons, and they're looking in a mouse where the interneurons that express the 5-HT3A receptor are the neurons that they label. And it's important, at least for thinking about it, that you realize that it's not just layer 1 that's labeled in this prep. So although they can't image their voltage sensor that deep, the blue light is definitely stimulating interneurons in deeper layers that express the construct. It just has to. Whether that's a confound that undermines their data, I highly doubt it, but you should just mention that. They're not using a line that's specific for layer 1. Even better, we know from Bernardo Rudi's lab that layer 1 has 4 different types of cells in it. And so they could try to break this down better, but they don't. They just sort on cells and effect. So it's not really clear to me if this sorting then means would reveal a cell type specific set of cells that react this way or not. Their model is just, you know, interneurons are connected and they inhibit each other, and the conductance model is enough, but we'll get there, sorry. So figure 2 is just sensory input and voltage monitoring with soma archon. And so they show stimulation and soma archon, and then they show in awake and anesthetized animals what the signal looks like in these 5-HT3A interneurons. And you can see that they spike singly and fire bursts when they're awake. So anyway, this is the activity that they see. So now what they're going to do in figure 3 then is try to dissect this circuit, and by dissect what they mean is that they're going to, holy gigantic terrible driving, holy crap, and you're going to go there, holy crap, you turn to U-turn in an intersection. That is completely and total, thank you sir, completely and totally illegal. So figure 2 shows anesthetized and awake animals and the results of sensory stimulation, i.e. flicking of the whisker. The next figure is going to dissect this interneuron network by depolarizing the network and see what happens when they flick the whisker, with the idea being that if the circuit is active, what happens. And so that's what they do. And what they see is that, first of all, they see some spiking and some inhibition following the spike. But more importantly, if the cell has been depolarized and is in the middle of a spike train, when they flick the whisker, they are able to see an inhibitory response that doesn't show up at baseline when there is no spiking. And so they make the hypothesis that during the depolarization, the conductance for the GABA chloride current changes, and then you see a GABA event that didn't really have much of an effect physiologically when the cell was at rest. And they do spike triggered averages and this kind of thing to show you that that's what they're seeing. And then the title of this figure is awake, but actually H, or at least the last figure in this where they show where they try to pair the depolarization with a whisker flick or without a whisker flick, and then do a spike triggered average to try and look at those waveforms. That's done in anesthetized animals. You can see that in the figure legend, but they kind of mislead you with the title of the figure and saying that it's in awake animals, because the last part of this figure is in anesthetized animals. So we see this inhibitory stuff and we only see it when the cell is depolarized, and so now what they're going to do is monitor cells in the middle of the field of view, and then at the same time, using clever light tricks, they're going to stimulate a surround ring. And again, they're stimulating inhibitory cells, right? And so what they're going to do is try and replicate this observation that they just made. They're going to use the blue light to stimulate the cells in the middle, and then I use a separate ring of blue light outside of these cells to stimulate interneurons in the surrounding neuropil, and see if they can recapitulate the observation that they made in 3, which was that when the target cell is depolarized, that it reveals an inhibitory signal coming from the surround interneurons. And you're an idiot. And so that's what they're going to do here, is stimulate that surround and see that when they do that, they see the exact same kind of inhibitory signal that they observed in figure 3. And then even more importantly, if they titrate the light in that surround circle, what they see is that the size of the inhibitory pulse is related to the amount of light that they use. So that also suggests that they are stimulating the interneurons in the surrounding neuropil and evoking the inhibition with the light. And it seems to be recapitulating the observation that they made during sensory stimulation. So then, of course, the flip side of this is obvious maybe, but instead of doing the ring of excitation to try and drive the inhibitory neurons in the surrounding neuropil, they're going to put the eye optopatch construct in, and when they stimulate the ring, they don't expect to see the inhibition then, because they will be hyperpolarizing the source of this inhibition. So now they're going to flick the whisker and see if they see the inhibition. Now the problem is that when they use this eye optopatch, they can't depolarize the neuron to create the effect that they saw in figures 3 and 4. And so the only thing they can do is get a single spike. And then with the spike triggered average, what they see is with the surround inhibition on, when they stimulate the whisker, the after spike waveform is essentially excitatory. Whereas when they aren't inhibiting the surrounding neuropil, the inhibitory signal after the action potential is at least somewhat visible. It's by far not perfect, but there's a difference between these waveforms, and so the authors claim that, you know, this is the result. And I'm not arguing with the result. I'm just suggesting to you that it's not as clean as you might imagine it would be. The effect is very subtle. Holy shit, I think I'm losing a pedal. a pedal. Shoot! So sorry about that break. Some bearing or something went in my pedal, and now I got massive clicking in my pedal. It's annoying. Wonder if I can fix that before the morning. So the last figure is one that I don't really understand a whole lot, because I don't really know what they're trying to show with it. Here what they do is they record from these interneurons, and they show that if they puff the eye of the mouse, that they can get a a general increase in excitatory inputs to these interneurons. So they spike a little more. And then they show that they can block this increase in excitatory inputs with a nicotinic receptor blocker, which is great. But then in figure 7 they make a conductance model and show with the conductance model that that acetylcholine can create a scenario where all these interneurons inhibiting each other end up with a winner takes all kind of thing where one spikes. Thanks for joining me on my ride. That was a new paper from Adam Cohen's lab showing the first All Optical Electrophysiology. It's a nice paper about soma-centered archon, which is their version of a genetically encoded voltage indicator. They show that it works pretty well. They show that they can depolarize and hyperpolarize neurons during recordings, and then they use this this toolbox to try and discern something going on within the inhibitory network of layer 1. If you want to learn more about the inhibitory network of layer 1, you need to go to Bernardo Rudi's lab, and you need to go to Stephen Williams's paper from 2014, wherein the details of this circuit are much more flushed out, and so then you can see better the sort of weaknesses that this paper has in terms of addressing the actual details of layer 1. Right now they've left it at an inhibitory network that that during depolarization can allow, through conductance changes, a amplification of GABA responses. This is definitely different than Stephen Williams's circuit, where he shows more complex cholinergic effects through nicotinic and muscarinic receptors. Nonetheless, this is part of that same circuit, and trying to figure out how this circuit works is going to require the integration of both the in vitro and in vivo data. The in vivo data is very nice, and it is proof of principle that this can be done. There are a couple caveats. The first one being that I'm guaranteeing you that Adam Cohen's lab chose layer 1 because that's the place where they get the highest signal to noise ratio. They show that in figure 1, that if they go much deeper than layer 2, 3, they're really at 50-50, and at that stage you're not really doing physiology anymore, and you're not making a good enough recording to where you're going to get anything. So there's still a lot of limitations to this sort of technique, but in terms of first pass probing of something, I think this is really nice. We did learn a lot about what's possible and what's likely to come in the future in terms of All Optical Electrophysiology. Thanks for joining me on my ride. If you liked it, like and subscribe, and I'll see you next time on JC on a Bike.